Students, this is the next topic. What is cell division? That is mitosis and meiosis. We are going to see about in this cell biology what is mitotic division and what is meiosis division. So we'll see what is a cell division. You can see here the diagrammatic representation what I have given here. Okay, so cell division is a process by which a parent cells divides into two or more daughter cells. Here a parent cell divides into two or more daughter cells. So there are two main types of cell divisions, if you can see here, mitosis and meiosis. So both the processes are essential for the growth and development and reproduction in living organisms. So both these processes are very important for the growth, development and reproduction in living organisms. Let us move to the next one. So we will see what is mitosis first. So this is a type of cell division, as I said earlier, that produces two identical daughter cells, okay, which produces two identical daughter cells. So it is essential for the growth, development and repair of tissues in multicellular organisms. Very important for the development, growth and repair of tissues. So the process of mitosis involves several stages. If you see here, prophase, metaphase, anaphase and telophase. Okay, so these are the different stages here what I have shown here in the diagrammatic way. Let us move to the next point, that is what is prophase in mitosis. You can see here this is the diagrammatic representation of what is prophase in mitosis. I told you all, if you see in the previous one, different stages are involved. First stage we are going to discuss now, what is prophase. So during prophase the chromatin condenses into invisible chromosomes. Okay, it condenses in the chromatin here, okay, condenses into invisible chromosomes. Okay, so the nuclear envelope breaks down and the spindle fibers begin to form. The nuclear envelope will be breaking and the spindle fibers formation will take place. So the centrioles move to opposite poles of the cell. The centrioles will move to the opposite poles of the cell. So next stage we are going to see what is metaphase in mitosis. This diagram represents the metaphase in mitosis. So here in metaphase the chromosomes line up along the equatorial plane of the cell. You can see here. Okay, so the spindle fibers attach to the centromeres of the chromosome. Here, this ensures that each daughter cell receives equal number of chromosomes. Each daughter cell will receive here equal number of chromosomes. This is the metaphase stage where I am showing in the diagrammatic way. Next one, anaphase in mitosis. Anaphase is the stage where the sister chromatids separate and move towards the opposite pole. Here you can see in the diagram. So the spindle fiber shortens, pulling the chromatids apart. It shortens and it will is how here is pulling the chromatins apart from where here it is closed and it is pulling here in this diagrammatic way. So this ensures that each daughter cell receives a complete set of chromosomes here. Next one is telophase in mitosis. So here this is a diagrammatic representation of telophase in mitosis. Telophase is the final stage of mitosis where the chromosomes decondense back into chromatin. This is the final stage. Here the nuclear envelope reforms and around each set of chromosomes. The cytokinesis, the division of cytoplasm occurs resulting in two separate daughter cells. Okay. Next one we are going to see, now we finished earlier about mitosis, now we are going to see about meiosis. What is meiosis? Meiosis is a type of cell division which produces four daughter cells with half of the number of the chromosomes as the parent cell. So this meiosis is essential for sexual reproduction and genetic diversity in organisms. So meiosis involves again two rounds of division, that is meiosis 1 and meiosis 2. So if you see here, prophase 1 in meiosis. Prophase 1 is the longest phase of meiosis, I should say, where homologous chromosomes pair up here and exchange genetic material through crossing over. This represents the diagram. This process increases genetic variability among the daughter cells. Here what happens, the nuclear envelope breaks down and the spindle fibers begin to form. Next, metaphase 1 in meiosis, if you see, in metaphase 1, the homologous chromosomes line up along the equatorial plane. This ensures that each daughter cell receives a unique combination of chromosomes. Here in the diagram, see, each one will receive a unique combination of a chromosome. This diagram represents. Okay. Next one, we are going to see anaphase 1 in meiosis. Anaphase 1 is a stage where the homologous chromosomes separate and move towards opposite poles. Okay, here the homologous chromosomes will move towards their opposite poles. So the spindle fibers are shortened, pulling apart, and this ensures that each daughter cell receives one chromosome from each homologous pair. Here you can see the diagram which represents here. Next one, we are going to see telophase 1 and cytokinesis in meiosis. So what is this telophase? Telophase is the final stage in meiosis where the chromosomes decondense and the nuclear envelope reforms. Okay, so cytokinesis divides the cell into two daughter cells, each with a haploid set of chromosomes. This diagram represents the reduction cell division. This process is followed by meiosis 2, which is similar to mitosis, but with haploid cells. Here it will be there. If you can see here, the next one, prophase 2 in meiosis. Prophase 2 is the first stage of meiosis 2, where we have seen meiosis 1 and now we are going to see about meiosis 2. As I told you, there are two divisions in meiosis also. So meiosis 1 we finished. So here the chromosomes condense back into visible structures. There is no crossing over in meiosis 2 and the homologous chromosomes have already separated in meiosis 1. So if you see here, metaphase 2 in meiosis, if you see, it will be there, the spindle fibers attach to the centromeres of the individual chromosomes. Here in this stage, it ensures that each daughter cell receives a complete set of chromosomes. So anaphase 2 and telophase 2 in meiosis, if you see, anaphase 2 is a stage where the sister chromatodes separate and move towards the opposite pole and the spindle fibers shorten, which will pull apart the chromatids. Telophase 2 is the final stage of meiosis where the chromosomes decondense and the nuclear envelope reforms, which results in the formation of 4 haploid daughter cells. So why is so important of cell division? We will see now. Cell division through mitosis and meiosis, I told you all already, it plays a crucial role in growth, development and reproduction in living organisms, and also it will help to maintenance of somatic cells chromosome number. Understanding of this cell division of mitosis and meiosis plays a vital understanding of evolution or complexity of life on earth. Genetics helps in many fields. So with this, we finish the explanation about cell division, mitosis and meiosis. If you see, please go through this explanation and make a note of it.